And we're back here at the American Dairy Science Association meetings. This session has a couple themes. One is Brazil. We have two guests from Brazil, and the predominant theme is Virginia Tech. So we've got three people here that spent some time at Virginia Tech. So that's kind of cool. We're going to be reviewing a paper by Yumi Taguti. Did I say that right?
Yeah.
Close enough? All right,
Yeah,
very well.
good.
And she's an undergraduate. She's visiting from Brazil. And she presented a paper. Was it a paper or a poster?
It's a poster today, yeah.
Yep. Called Linking Amino Acids to Milk Fat Synthesis. So, welcome. Can you tell us a little bit about how you got the idea for this research?
Yeah, sure. So I think the first thing we need to understand is how milk fat is important, because milk fat is what drives the milk price in the industry. So knowing that the milk fat yield in cows supplemented with amino acid has changed, we had like in vitro studies, previous in vitro studies about how the mTOR pathway acts behind this milk fat yield changes, and this was realized that this was conducted in mammary epithelial cells in supraphysiological levels. So what our goal now is to understand how is the relationship between the essential amino acids and the milk fat synthesis and physiological levels.
Oh, very interesting.
Yeah.
And I see you brought a guest with you today. Is this your professor?
Yes.
Yeah, Izabelle Teixeira.
Perfect.
Close. All right.
Excellent.
Now, yeah, they're confusing me just a little bit because you're no longer at Virginia Tech. You're currently at the University of Idaho, but you were there doing a sabbatical.
Yeah, it was in 2017. I was a faculty in Brazil at Unesp, and then I went to do a sabbatical at Virginia Tech.
Okay.
When we were discussing, Mark Hennigan and I, about the project, the possibilities, we came out with this idea just to evaluate the role of the essential amino acids on the milk fat synthesis. And the very first experiment study in this sense in our group, I mean, my group and Mark Hennigan's group, was in that time.
Yeah.
So it was nice, and we could figure out some amino acids that were playing a role because we were omitting amino acids on the media on the epithelial cells, mammary gland. So, and we could see that when we were omitting some amino acids, it really shut the de novo milk fat synthesis. So I stayed 2 years. It was a 2-year sabbatical. I went back to Brazil, and currently I'm at University of Idaho. I just joined University of Idaho.
Okay.
So that's a long story, and now join me through those all changes.
Yeah. Well, we're happy to have you here with us today
Thank
here
you.
at the Real Science Exchange. My co-host, Carrie Estes, once again.
Again.
Yeah, you've been here your third time. So I'll hand it off to you. I think you got some questions.
Okay, yes. So I'm not an expert at all in cell culture. I'm just going to set that up right now. But I was curious about the stage of lactation that you took the mammary tissue from. What stage were they at?
Okay, so I think the most important thing that we considered when we were taking the tissue samples was to get from a cow that is a high producer animal, but I think mostly in the mid lactation.
How did you define high producing? I'm sorry.
High producing for in the farm that we're working currently, high producing will be around 50 kilograms of milk per day.
That's high.
During 2 milkings. But I was not involved in the cell isolation process because I was not here yet. I think maybe Izabelle can tell you more about this part of the process.
Yeah, so we did some because we had some papers back in that time, and I think you joined some. I sort of overlap with Carrie at Virginia Tech. So anyway, every time you were reading papers about isolating cells, it was not very clear. So we were able to establish at Hennigan's lab a way for isolating cells. So I started doing that with biopsy samples. That was nice. That's why we were able to get like middle lactation because we want to be sure that the cells were able to secrete milk components, because that was important for us and also for other studies that Mark were running at the lab at that time. After that, after I went back to Brazil, Alex Burberry also refined a little bit more the technique. But just being direct to your question, so mid-lactation is good, and we want to be sure that that cells are secreting milk components.
Yeah. 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 It becomes pretty important then.
It
yeah.
is.
Sure.
Yeah.
Sure.
Yeah.
And with those cells, we were able to check that they were secreting protein and fat.
Okay.
So we could address different questions with the same cells.
So then once you have your cells and you're applying the treatment, how long do you let that treatment work on those cells before you end up taking your sample?
So what we did, we started with 23 hours of starvation on the cells, so they would not get overloads. And after that, we applied the treatment for 4 hours. So we were using labeled acetate as a tracer, C13. So the period that we need to let the treatment stays in the cells is more about it's related to the period that the acetate enrichment increases and it reaches the stability. So Izabelle conducted a study before to know when was the increase. When was the best time the enrichment of C13? So after this research, we could choose 4 hours for the treatments. Yeah.
Okay.
Yeah, it depends on the kind of questions, right, Carrie? Because, for example, if you are doing Western with those cells, we cannot go, if you want to do to check the phosphorylation, generally we go up to 1 hour and a half maximum because we want to see the phosphorylation. That is a very fast process. With the isotope, because we want to see the enrichment of the cells with C13, we want to get is still up the curve, not reaching the plateau. So we chose 4 hours. Generally, that's what we're getting the best. But Yumi also used with a second trial that she's running right now at Virginia Tech. We just figured out that perhaps it would be better to have the cells exposed a little bit more. So she exposed the cells to 6 hours, right?
Yeah, 6 hours this time.
Yeah. So as we are, because sometimes just can get confused, is it enough time? Because we are focusing on mechanism and really understand the mechanism. So we need to address those questions accordingly. That's why we are using a short period of time.
Yeah, makes sense. Makes sense. So you did a lot of this work, very interesting work in vitro. What about in vivo? Do you guys have plans to move this into the cow?
Yeah, we do. So we had 2 in vitro experiments until now. In the first one, we were analyzing the individual effects of essential amino acids. In the second one, we could see it's still an ongoing experiment, but we can see the different levels and interactions between the amino acids that show most effect in the first experiment. And after that, we are moving on now for the third experiment that I'm doing here. It's an animal trial. So in this case, we are infusing the animals with the amino acids and trying to get an effect on the milk fat yield. So we still don't have any results about that, but yeah.
It's in the works.
Yeah, it's
Stay
working.
tuned. Okay.
Let's see.
Okay, very good.
So you don't have any results on that specific, but some of the previous results, can you talk a little bit about those and the conclusions that you may have come to?
From the animal trial?
Yes.
So,
No, well, I was even thinking the whole series of trials that you've done so far.
oh, the whole series of trials? Yeah, of course. So during the trials, seeing the results, we could see that omitting any of the amino acids, when we remove any of them, we do have an effect on the milk fatty acids. It does change something. And we could also see that some of the amino acids that I found that had the most effect on the milk fat yield was the same that Izabelle found on her previous research with supraphysiological levels. For me, I found lysine, methionine, isoleucine, and leucine being the ones that affect the most the milk fat yield.
Okay.
Yeah, I think that's what we got as big results right now. So those are the amino acids that we're using in the second experiment to analyze the different levels and the different interactions between them.
Very well. Very interesting.
Yeah, and also the ones that we're going to use on the animal trial, right?
Yeah, they're the same. Then
Yeah. And the idea is to see it, because at this point we already know that just addressing one amino acid time is not the best way because they interact, right? So she's designing the experiment in a way that we can see the interactions among the amino acids and see the amino acids as a group, not only as individual amino acids.
Mm-hmm. So going forward, what do you see as maybe some practical implications, and then maybe talk about some additional research that's going to be needed?
Yeah, sure. I think some practical implications. I think this knowledge is important that maybe in the future we can manipulate a diet in terms of amino acids to try to increase the milk fat yield. That would be really interesting for the producers.
Mm-hmm.
And I think the future research, we still need to explore more how the amino acids are interacting and which amino acids we should work together, which should be the groups so we have better results.
Very well. Speaking of the future, what's your future hold? You're now a visiting student here at Virginia Tech. What's next for Yumi?
You're putting me in a hard place here. Okay, so I think my future, I really want to go to grad school. I think about teaching in the future. So, yeah, I have an opportunity now, and I'm still open for opportunities. Yeah, that's it, I think.
Okay, very well. So anybody out there listening, Yumi's open to other opportunities. Very well.
And I'm in the line. You know? She already has an invitation
Now,
for
here's where
grad
I'm in
school.
trouble.
Yeah, that's my opportunity. That's my main goal. I'll be there.
You know, I'm convincing her to go back to Brazil, finish the vet school, and then come back to do the PhD at University of Idaho.
And I'm willing to, yeah.
There you go. All right. Well, listen, I've really enjoyed the discussion this afternoon. Izabelle, thank you for joining us.
Thank you.
Yumi, it's been great, and all the best to you. Good luck. Whatever you
Thank you so
decide.
much, yeah.
All right.
We'd love to hear your comments or ideas for topics and guests, so please reach out via email to anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars continues with ruminant-focused topics on the first Tuesday of every month, monogastric-focused topics on the second Tuesday of each month, and quarterly topics for the companion animal segment. Visit balchem.com/realscience to see the latest schedule and to register for upcoming webinars.